Okay, I'm going to take roughly, hi, this is Nick Cap, and this is the stem cell class, and I'm going to take roughly a few minutes to kind of talk about this particular book, Basics of Cell Culture, by Gulna Ashfar, who's an excellent teacher. We're actually going to cover up through roughly about page number 33 today, and I want to take roughly about 10 minutes to do it. So it's 11:52 right now. We'll go on there. Basically, you want to read about the basics of cell culture, and we haven't been able to do cell culture all that long. If we look at it here, Hayflick's limit was basically about 1961, and so the magic about cell culture is basically being able to use a newborn calf serum. With this, what I want you to do is I want you to be able to conduct yourself safely in the laboratory. So no eating or drinking, keep your hands away from your mouth, no horseplay, wash your hands before and after. If there's a laboratory accident, you definitely need to tell me about it. Friends and visitors really kind of can't come in because they do that. Closed-toed shoes and pants must be worn at all time. Cell phones turn off. And again, familiarize yourself where you are. Don't turn off your brain. You need to use your basically your skills and your common sense skills. We're not necessarily going to use laboratory notebooks in this class. We are going to be doing stuff on Google Documents, so we will be able to document that, so make sure that you are aware of those. We're not going to have that. The laboratory culture, we went over it, and I want you to know and read about and look at kind of what is a laminar flow hood and explain how those particular work. You need to know what an inverted microscope is and how they work. We have 4 of those in the class. We're not going to be using fluorescent inverted microscopes, but that is something that we can use. Centrifuges we'll use kind of all the time. We didn't use one the other day, but we will be using those in our class. The main thing with a centrifuge is getting it balanced. If it's balanced, then it will work pretty well. If it's not, then it's not going to work well, and be aware of that. And so this is looking at that balance. The second thing is know how a CO2 tank works. And again, we're going to be using, why we use a CO2 tank is because we have this thing called bicarbonate buffer in the cells. And again, this interaction stabilizes the pH at around roughly 7.5. And so we see in the presence of water, which all our media has, we have this ion being made, and actually this substance being made, and it actually converts into the bicarbonate, and actually we use this to maintain the pH of the cells. And so when our cells are pretty happy, they're around 7.4 pH, and they're that kind of nice red color. If they start to get too purple, then they're getting too alkaline, their pH is going too high. If they're getting too yellow, then they're getting acidic, and we don't want to have that happen as well. We will be using 30 degrees cell water baths in order to change the temperatures. We use lots of refrigerators and freezers. The refrigerators are basically set at 4 degrees Celsius. Freezers are usually set at -20. We have a number of biohazardous waste containers. Again, these are expensive to get rid of the waste, so we don't want to use, we want to use them when we have to use them, or when it's proper to use them. So again, tips and everything else that touch are contaminated are put in there. But again, gloves that are sanitized after you're done with them don't necessarily need to go in there, and those can go into the regular waste. We will be looking at cell culture vessels. A lot of our cell culture vessels are disposables, and we'll look at that. And so again, here's looking at those cell culture vessels, and then we will be using these bottles over here, 6-weld plates over here. You need to know how the 6-weld plates are configured. Again, row 1, row so the rows and lines and columns and kind of things like that, and be aware of that. For the pipettes, it's very important that you know how to use a serological pipette, but basically we'll be using most of are the microbe pipettes, and so you need to be able to use those, and we'll talk about calibration of those a little bit later. One of the big things that you want to talk about, you want to learn about, is contamination. And contamination is basically an infection of unwanted materials into our cultures, and that could be another tissue culture, it could be bacteria, it could be yeast, molds, and all those kind of things. And what we do a lot in our laboratory is all the techniques that we have are basically to prevent microbial contamination. Our cultures have phenol red in them. The phenol red is what kind of gives it its cultures. If our cultures start to get acidic, that would mean our culture is getting a little bit old. Here's a nice little picture of Chinese hamster cells, and these cells are infected with the yeast, and you can actually see this. So these cells look a lot like our mouse macrophage cells inside of here, and we can see these other smaller cells inside there are the yeast, and we do not want that to happen. So we use this process that we call aseptic technique that will prevent that from happening. And again, how do we do that? Is by basically maintaining clean equipment. We wipe our materials down with 70% ethanol. That's the spray bottles that we have. Don't put your notebooks and stuff inside the hood. Don't basically put anything in the hood that you want. You want to try not to cough or sneeze, or kind of do it in your jacket. All solutions and reagents we start out sterile. So if something's sterile, it will remain sterile. If something's not sterile, it will unsterilize kind of everything else. Again, never use other people's medias. Be careful. Make sure that you label everything so it's not looked up. And so again, look at all this material, and you actually gave me some ideas of what to do. And so they have lots of steps, at least 21 steps. For microbial contamination, what you want to do is either squirt Sanasol or bleach on it and let it sit there for at least 10 minutes. Okay, and then that will decontaminate it, and be careful with that. Lab number 6, common laboratory practices. You always want to practice good aseptic techniques. Always wash your hands and clean your work before and after you're done. Always observe your cells before any procedures and afterwards if you can. And for most routine procedures, you may have to sacrifice extreme accuracy for the sake of speed. And it's better to be a little sloppy and fast than it is to be very meticulous, but it slows you way down. And we'll talk about this a lot in our class. So for your media, for the most part, we'll have the media already made for you. Make sure that you are using the proper media. When I give you some media, make sure that you put your name on the media. The media needs to be stored at 4 degrees. We typically will warm it up, but if you leave it in the culture conditions for a day or two, your media does go bad. So we talk about this. For routine cell culture and maintenance, feeding and subculture, what we want to do is we're going to subculture, and we're going to feed at least twice a week, and we will subculture at least once a week, and we'll talk a little bit about that. Our mouse macrophage cells are really great in that we do not need to trypsinize them in order to kind of move them. The first laboratory that you did, here's counting using the hemocytometer. Please read about this, and we'll cover this, and we'll do the counting kind of next week, and that's it. And then you guys actually did what you guys actually did is basically look at your sterile technique, and you guys did a good job on that. And so again, I wanted you to read through roughly about page 35, which is this page right here. And so basically you want to talk about counting the cells. All right, with that, I will leave you now.